The 100,000 handshake problem. The average human cell manages more than 130,000 unique protein-to-protein interactions every second just to keep you alive. If even one of these molecular handshakes fails or happens with the wrong partner, the result is often a metastatic cancer or a neurodegenerative collapse. For decades, drug discovery was a guessing game, because we could only see individual proteins, not the network. But yesterday, on May 20th, 2026, a new framework called DMG-PPI proved we can now map these social lives of molecules with over 90% accuracy. This is the moment biology stops being a list of parts and starts being a predictable circuit board. We are finally seeing the logic behind the alignment message passing system, breaking the sequence ceiling. Most AI models fail at biology because they treat proteins like simple strings of text. In reality, proteins interact through two contradictory rules: similarity and complementarity. Think of it like a puzzle. Sometimes pieces bond because they look identical, called symmetric motifs. Other times they bond because they are perfect opposites, like a lock and key. The breakthrough in DMG-PPI is a dual-channel architecture. It uses one pathway called alignment message passing to find the similarities, and a second channel called divergence message passing to find the opposites. By running these in parallel, the AI effectively sees the protein in 3D for the first time. This dual-view approach is what allowed researchers this month to identify hidden hotspots in proteins previously thought to be undruggable. This structural awareness leads us directly to the concept of protein social graphs, the LinkedIn for molecules. When you look at the May 2026 data, the most striking reveal is that proteins have high-order interaction patterns. They do not just bond in pairs. They form massive, influence-driven communities. This is essentially the social graph of life. Using a technique called mix-hop, the DMG-PPI model can predict not just if two proteins will touch, but how that touch ripples through the entire cellular neighborhood. In the recent study on Parkinson's disease, this network-level view allowed scientists to find a single protein interaction that was sabotaging the brain's energy supply. By treating the cell like a social network, we can finally identify the influencers. If you can target the high-traffic hubs in a disease network, you do not need to find 1,000 different drugs. You just need to disrupt the center of the graph. But precision requires more than just seeing the graph. It requires clinical-grade data, the 70% precision gap. The industry standard for predicting protein interactions has hovered at a frustrating level for years. Baseline models often struggle with noise or incomplete data, making them useless for actual clinical drug trials. DMG-PPI just shattered that ceiling with a 7% improvement in its F1 score over the previous best methods. Even when researchers intentionally perturbed the data or used incomplete sequences this month, the model remained robust. However, the real devil's advocate point is interpretability. Most AI is a black box. You get an answer, but you do not know why. DMG-PPI changed the game by pinpointing exactly which amino acid residues are driving the bond. It is not just telling you that an interaction happens, it is showing you the physical mechanism. This level of transparency is what will eventually replace the trial and error of the modern lab. The death of the magic bullet. We are witnessing the final shift from finding magic bullet drugs to engineering biological networks. The success of DMG-PPI and the dual-channel approach proves that sequence data alone is a dead end for the next decade of medicine. By integrating both similarity-driven and complementarity-driven pathways, we have moved from guessing at protein shapes to predicting the behavior of the entire human interactome. For the strategist, this means the value in biotech has shifted from owning a single molecule to owning the most accurate network map. If you control the graph, you control the intervention. We are moving toward a world of multi-protein complex design, where we do not just block a receptor. We reprogram the way a cell communicates. Watch for the first clinical trials using these dual-channel models for personalized oncology targets to begin in late 2026. The real indicator of success will be the first successful disruption of a multi-protein complex that was previously deemed impossible to target. Subscribe for more.